Good day, grade 12 learners. Welcome to today's Life Sciences lesson. My name is Sina Sekheche, and this lesson is brought to you by the Free State Department of Education in collaboration with Pagama Research and Development. Right, now we are meeting for the post-prelim revision. You have sat down and wrote your prelim examinations and you know how you did in those examinations. Now it's time to evaluate what is it that you can do to improve the marks that you got. So we're going to change the strategy now. You are going to assess yourself, analyze your results, and implement a new strategy that will work for you. So for today's lesson, we are not going to look at all the topics for paper 1. However, we are going to look at the big topics, or the topics that form or that have a high weighting in paper 1. So those topics will be human reproduction. So most of you are not doing so well in this topic, human reproduction, especially when it comes to the female reproduction with the hormones and all of that, up to pregnancy and implantation, fertilization. All of that, those questions, you don't do so well in them. So we'll check some of the questions that appeared in your Free State paper. Then we'll see where you went wrong. And also, we're going to look at the human's response to the environment. So this is the question that will have your nervous system, the brain, the eye, and the ear. So we're going to look at these two major topics for paper 1. So without wasting any time, you know the structure of the paper. You've seen the structure of the paper because you've written your preliminary examinations. Remember, those exams are preparing you for the final examination. So they are exactly as how your final examination is going to be structured, but with different questions. So now, by now, you should know where are you, or what level are you obtaining in life sciences, and how can you improve that. So you have time now to do the final last push for you to get that mark that you want. So the topic of human reproduction is a very huge one, and it is divided into the female reproduction system, the male reproduction system. So you see the female one, so you must know the diagram of the reproduction system. You must know the labels and the functions of the parts, and you must also be able to draw a diagram of the ovum. With the male, you must know the diagram as well, the labels, you must know the side view and the front view. Then the functions of the parts and diagram of a sperm cell, very important. Then puberty. What is puberty? This is just a small section that you do not do so bad in it, but menstrual cycle. Now, this is where the biggest challenge comes. Menstrual cycle, events occurring in the ovary, the ovarian cycle, and the uterus, the uterine cycle. So this ovarian cycle, so you are not able to follow the flow of hormones, what happens in the ovaries, what happens in the uterus, what happens in the pituitary, which hormones are released and when, and what will be the effect of those hormones on the whole menstrual cycle. So we're going to look at an easier way to remember the flow of those hormones. And then the hormonal control, this is what I'm talking about. These 4 hormones, there are only 4 hormones for you to remember. The key thing is to remember when is it released, by what is it secreted, and for what reason. So you need to know all those and make it follow the sequence of events. And then the development of the fetus. Most of you, when it comes to this one, you just rush through it, and there may be questions in your preliminary question paper. There was a question on the developing embryo in the uterus, and that may be a surprise to some of you because you only end up here, and then this one you just think that you know, but you don't go into details as to the blood vessels and the placenta and all the layers covering the embryo up until it becomes a fetus. So please make sure that you know all of this. Right, without wasting any more time, we're just going to go through this as revision, just to recap, because I want us to deal with direct questions today. I want us to have more time to dissect the questions on these difficult questions. So remember the male reproductive system. for you to be able to remember the diagram and its labels. Diagram and labels. I'm going to show you today that it's very important for you to remember a diagram and its labels. So you must follow the order. If a diagram requires you to start from outside to the inside, you do so with the labels coming in layer by layer up until you come to the innermost part. And then if a label requires you, or a diagram requires you to start from inside to the outside, please make sure that you follow the sequence, or you follow the order in which these parts are working together. That will help you a lot in remembering the parts and their functions. So we start with the male. The male one, it says it has a pair of testes. When they say a pair, it means two. So meaning there are two testes situated where? In the scrotum. So you're going to go to your diagram and check. This is the scrotum. Because this is a side view, you only see one testis. And then on top of the testis, you see there is an epididymis, and that epididymis will then lead to the vas deferens, the ejaculatory duct, and the urethra. By doing so, you are following the movement of the sperm. Where is it going to travel? So it's going to travel from the testis here to the epididymis here, where it's going to be stored until it matures, and then it's going to travel through the vas deferens to the ejaculatory duct, up and to the urethra, then to the outside. So you see, by doing so, by following these labels, you are able to even trace how sperm is going to travel. Then the male reproductive system does not only have a passageway. It also has glands that are assisting. So these, we call them accessory glands. So the accessory glands that we have 3 accessory glands. So what are they? You have the seminal vesicle. This one, you will remember that it looks like a leaf. Every time when you see the one that looks like a leaf, you see it has rough edges like this. It is your seminal vesicle. And then underneath the seminal vesicle, you will find a bigger oval to a circle, or a circle to an oval, but it's a little bit bigger than the other one that we're going to talk about. So you will know that that one just under the seminal vesicle will be your prostate gland. And then the last gland that you're talking about, so these are your accessory glands, all of these here. So the smallest one will be your corpus gland. How are you going to remember them? Small one is the corpus gland. The one that looks like a leaf is the seminal vesicle. And then this last one is your prostate gland. So those are the glands, and you will see their functions as we move. And then you have the penis. So the penis is just this muscular layer surrounding the urethra. So you see that the muscles around that. So that is your penis. So you are done with your male reproductive system. So let us check the glands. What are they secreting? The glands, remember the seminal vesicles, they produce seminal fluid to transport the sperm cells and fructose for nourishment. So to nourish the sperm, it has this type of sugar here, which is fructose, and it also has a fluid to help the sperm to move. So it will secrete that into that passageway to help the sperm to move, to the transport of sperm cells. Then the prostate gland, this one is the one that is going to secrete what? A milky, slightly alkaline fluid that is going to neutralize the acidity of the vagina. Remember, this sperm needs to travel through the vagina. So if the vagina is too acidic, then the sperm might denature, or it might be harmed. So the prostate gland will make sure that all this secretion of the semen has an alkaline fluid that is going to neutralize that acidity of the vagina, where the sperm is going to travel until it reaches the ovum. And then the corpus gland, that is your last gland. This one also secretes a fluid for greater motility of the sperm, so for greater movement, so it makes the movement even faster. So that is your motility. So all in all, these secretions of the prostate and the seminal vesicle, they nourish the sperm and then enabling them to swim, and then they provide also protection. Then you are done with your male reproductive system. There's not much for you to to remember here, but you must just remember how sperm cells are formed here in the testes. We will talk about that just now. So I would like us to look at the questions, but this time we are going to do the activities very differently. Each time when you come across a diagram, you must take your pencil and put on the labels on the diagrams, and then go to the questions. So you start by labeling, and then later on you do the questions. By labeling, then you can remember even the functions, or you can even remember the structures. And then when you get the questions, it's easier for you to just remember. So I would like to give you are not answering the questions. Remember, the questions, we'll do them together. What you are doing right now is for you to label. I would like for you to label all the parts that are not labeled there. So I'm going to give you just 1 minute. 1 minute to put in all the labels, and then we do the questions together. So I want you to label this in your head or on your script book just for 1 minute. Right. By labeling this diagram, I think it puts you in a better position. It gives you confidence to be able to expect questions or any questions coming thereafter. And you can even get 70% of the answers by just doing so. So please make sure that you apply this strategy. When the invigilator is passing there while you are writing, I would like for them to see you labeling. Unfortunately, I won't be there to monitor that, but I would like each and every one of you to label their diagrams as they receive any question. Right, so let us delve into the questions then. 1.1, give the letters of all the structures that are involved in each of the following. Now, all, so they are not limiting you. Then you must check, what does the question want? Letters, not names. So you must answer according to the question. This is one of the mistakes that you make, that you might just want to give the name, whereas it was asking for letters. Please read your questions carefully. Now, which letters or which structures are involved in the production of the fluid part of semen? So which ones are involved in the production of the fluid part of the semen? Remember, the semen will have all the liquid of the sperm and all the liquid of the secretions. So which parts are those? So you have your A, you have your B, and your C. All of those are involved in the production of the fluid part of semen. And then the transportation passageway of sperm and semen. So where does sperm travel? So which parts are involved in the traveling now of sperm? So already you have your answers there. So those parts will be your D, and you have your E, also your urethra and your ejaculatory duct. And then name gland B. You're going to go check your glands. You know this one is the one that looks like a leaf, which is A. B is the bigger circular one or oval one. And then C is the smaller one. What is the name of B? B will be your prostate gland. And then the hormone that is produced by the testes, that brings about the development of male secondary sexual characteristics. So what is that hormone? That hormone is testosterone. And you know that it is produced here in the testes. So that is the question that was in the 2022 prelim. I don't want us to do the next question. However, you have this worksheet with you. You can complete the rest of the question. But it was important for you to answer questions based on the structure and diagrams. So I want us to move to the next one, to the next question. Right. So this one was a question that appeared in the paper that you've just written now in Free State. So it says, first of all, you must just label. You label, you label, you label. Even if there are no labels, just write somewhere there on the side what you think this is, what is this, what this is, what this is, what is this, and then all of these. So you must label. Right. And then it was saying to you, identify part A. Part A, we've spoken about it a lot today. So that is your, then you have the answer. 2.5.2 says prostate cancer affects men and the risk increases as men get older, which may lead to surgical removal of part B. So part B is this one. So that is the one that causes the prostate cancer. And then explain how the removal of part B may affect male fertility. So if they remove this part B, how is this going to affect this male's fertility? Then describe the process of spermatogenesis. I would like for you to just take note of this question because when we deal with it later, I will show you how to answer this question. So first of all, what is part A? Part A is your seminal vesicle. And then how will this man's fertility be affected if part B is removed? This is how. An alkaline fluid will not be secreted. You start by the function. Remember, the function of the prostate is to secrete that alkaline fluid to neutralize the acidity of the vagina. So because the prostate is removed, that alkaline fluid is not going to be removed. You get 1 mark for that. And then to neutralize the acidity of in the urethra or in the vagina, because because of copulation, therefore the urethra will also be in contact with the vagina. Then sperm cells will denature. Yes, because why? Because of the acidity of the vagina. And then, or they will die from the abnormal sperm cells, and then the decrease in male fertility will therefore occur. So you need the secretions of this prostate gland to form part of the semen to make sure that this whole process takes place without the sperms being denatured. Then describe the process of spermatogenesis. This is a question that I said to you, please keep it for later. Then we will do it just now. Right. Then we move on to the female. So this is the female reproductive system. You can see this is the side view, and then this is the front view. The front view, you will mostly get questions with the front view, but you can also familiarize yourself with the side view and check where all these organs or how they would look like if you are looking at them from the side. Right. But let us look at the front view. So what does the female reproductive system consist of? It consists of a pair of ovaries. Remember, we said because it says a pair, it means two. So here are the ovaries. You start here. Here are the ovaries. And then the fallopian tubes are leading from each ovary. So you see the fallopian tube leading from the ovary, fallopian tube leading from each ovary. And then they join into the uterus or the womb. Now we are here. This is our uterus. You see? Our uterus, and then the lining of the uterus inside is called the endometrium. And then the vagina, you have the cervix here, just at the opening of the womb or the uterus is called the cervix. And then leading from the cervix, it will lead into the passage called the vagina. It's a muscular passage called the vagina. And then, or it can also be called a birth canal. And then the external opening, which is the outside of this female reproductive system, which will be the vulva. So these are the parts that that the female reproductive system comprises of. But there is a lot that is happening in the ovaries that we need to look at. So we will delve into all those processes. Right. So just for the diagram, for you to remember, I will give you again 1 minute for you to label all the structures that you see there in the female reproductive system, and then we will continue with the questions. Do not answer these questions that you see, just label. Label for 1 minute. Right. So I think as you are practicing this skill, it's going to get into your head. You're going to keep on practicing and then you're going to master it. Right. Then this question was coming from the paper that you've just written. And it was asking you to show you, okay, the diagram below shows the female reproductive system. Now, identify part A and C. You see that you've already done that by just labeling these parts. So it's easy for you to just write now the labels without even having to scratch your head about it. Now, give the letter only of the part where each of the following takes place. So take note of the question, what is it asking? So where does meiosis take place and where does fertilization take place? So let us look at the answers. Your A will be your fallopian tube, and your C, what will be your C there? Your C will be your cervix. Then where does meiosis take place? It takes place in part, remember they only just want the letter, in part B, takes place here in the ovaries. And then where does fertilization take place? The fertilization takes place in the fallopian tubes, there in A. So it's going to be easy for you to write those if you know the functions also of the parts. Remember, you don't just study the parts, you study also their functions. For you to get at least that 30%, that 40% mark in life sciences, you must know all your labels and their functions. Right. Continuing, state 2 functions of part E. So this is part E. Then you must have there in your label that part E is the uterus. Please write the scientific name. I know it's also called the womb, but just write the scientific name, which is the uterus. And then 2.4, name the gland which secretes nutrient-rich fluid for the sperm to reach part A. So which gland is that? You're going to name the gland. So let us go back to the functions of part E. What is the functions of the uterus? The uterus is responsible for the protection of the embryo. Remember, the embryo is going to attach here on the uterus. And it also, from implantation to birth. And it also provides space for the developing fetus. So the fetus is going to grow from this uterus, the womb, what you know as the womb. This is where the fetus is going to grow. So it provides space for that growing fetus. Now, name the gland which secretes nutrient-rich fluid for sperm to reach part A. So we want sperm to come here. So, and remember here, it is acidic. So which gland is going to release that nutrient fluid? It is the seminal vesicle. Now, it did not have anything to do with the acidity, but they wanted the nutrient rich. Remember that fructose that we've spoken about. So it is secreted by the seminal vesicles in the males. in the male reproductive system, not in the female. So they are intertwined because this is where fertilization takes place. This is how fertilization takes place. And then puberty. What you need to know about puberty. What is puberty? Puberty refers to the age at which sexual maturity occurs and sex hormones are produced, which leads to secondary male or female sexual characteristics and gametogenesis. And you know gametogenesis in the males, it's going to be called spermatogenesis. In the females, is called the oogenesis. So this is where the formation of the ovum is occurring. This is where the formation of the sperm is occurring. Together, we call it gametogenesis because gametes are your sex cells. So in males, sex cells are sperms. In females, sex cells are ovum. Right. So puberty brings all of those changes. Then it starts where in the males? It starts in the pituitary gland where FSH is going to be secreted. Also in the females, the pituitary gland is going to secrete what? FSH. Now this FSH is going to stimulate the development of the follicles. Here in the males, it's going to stimulate the testes to produce sperm. And then also LH from the pituitary gland is going to stimulate the testes, because you might ask yourself, when do the testes know when is the time or when is the right time to start producing sperm? At the right time, when a boy child reaches puberty, the LH is going to send that message to the testes to start now producing testosterone. And then that testosterone, remember, this is the male hormone, is going to give a male what? Their secondary sexual characteristics. You know all those characteristics that are visible, the physical characteristics that you went through with your teacher. And then in females, this LH is going to stimulate the development of the corpus luteum in the ovary to produce progesterone. Then it is not responsible for the secondary characteristics in females. Remember, estrogen causes those secondary characteristics which are produced in the follicles. So estrogen in the follicles of the ovary, that is the one that causes the females to have the secondary sexual characteristics from that age. For some females starts from age 7 to age 19. For males from age 9. So it depends on the development of a person. But all in all, this is puberty. You must know that in females, the secondary sexual characteristics are under the influence of estrogen, and in males, the secondary sexual characteristics are under the influence of testosterone. Then we move to gametogenesis. What is gametogenesis? It is the formation of gametes. What are gametes? They are sex cells. In males, the gametes are sperms. So how are these sperm formed? So remember that question that I said to you, please leave it for now. Then after discussing this, you will be able to answer that question. Right. So under the influence of testosterone, you must tell us what brings this first. So testosterone is the one that is going to start this process. A diploid germinal epithelial cell. So where do you get this diploid germinal epithelial cell? From the lining of the testes. From the lining of the testes, you find the germinal epithelial cells, and they are diploid. And then in the seminiferous tubules of the testes here. So you must be specific where. In the seminiferous tubules of the testes. What is going to happen to them, to these diploid germinal epithelial cells? They are going to undergo meiosis and form what? 4 haploid sperm cells. You don't need to explain it in detail in what. So these are only the 4 or how many? 5 bullets that you need to remember. Under the influence of testosterone, a diploid germinal epithelial cells in the seminiferous tubules of the testes undergoes what? Meiosis to form 4 haploid cells. As simple as that. So this is how gametes are formed. So these haploid sperm cells are our gametes. Then this is our gametogenesis. And how is this gamete of the male, how does it look like? If you take a look at it, you see on this diagram here, this is the diagram of the sperm. So let us take a look at the diagram. The diagram is divided into 3 sections. Remember, you have the head, the middle piece, and a long tail. Please emphasize the long tail. It's not just the tail, it's a long tail. And then which parts comprises of the head? And remember, the head is the only part that enters the ovum during fertilization. The head has an acrosome. Here is the acrosome. It contains enzymes to digest the walls of the ovum for fertilization. Remember, the ovum also has its walls. It has the jelly layer. It also has a cell membrane. So this sperm cell needs to be able to penetrate into the ovum until it reaches the nucleus. Now, the acrosome of the sperm enables it to do that because it has that enzymes that will be able to digest those walls of the ovum. Then it has a nucleus which contains 23 chromosomes, and these 23 chromosomes carries genetic material. 23 is made by 22 autosomes or 22 normal chromosomes, ordinary chromosomes, and 1 sex chromosome. So it will depend whether it's an X or a Y on chromosome number 23. So in this nucleus, it is haploid. You only have 23. It is not diploid. You have half the genetic material because it has now formed here. Remember, we said 4 haploid cells. So because it's haploid, it has only 23 chromosomes in the nucleus because we want this nucleus, when it fertilizes the ovum, the 23 of the nucleus of the sperm cell and the 23 of the nucleus of the ovum fuse together to form a haploid zygote. Then that will develop into a fetus later on. Right. Then you find a centriole, and then on the middle piece, you find mitochondria. Then this mitochondria provides energy for swimming. It provides the sperm with energy for swimming. And then the long tail that is a flagellum. Flagellum it is moving or used to swim. So it is used for swimming. And when you draw this, please make sure that your tail, you see the tail is longer than the previous 2 parts. Emphasize the long tail. So even if you can draw your sperm and then draw a tail that will fill your booklet up to the right margin, do so, because then you will be emphasizing that this is a long flagellum tail. That is how you draw your sperm cell. Then moving on. Then we have oogenesis. This one happens in the females. So you remember it said that you must be able to draw also the ovum. So this is a diagram of an ovum. Right. So how is this ovum formed? First, before we start looking at it, how is it formed? Diploid cells in the ovary undergo mitosis. Please take note. Mitosis first because we want them to be many, to form many follicles. So a diploid cell in the ovary. So in the ovary, there are cells there, and then those cells are diploid because they are full cells. They are going to undergo mitosis so that they produce what? Many follicles. Then what's going to happen with those follicles? At the start of puberty, remember, all those hormones need to now come and activate the process. At the start of puberty and under the influence of FSH, very important, under the influence of FSH, one cell in the follicle grows and undergoes meiosis. So because we produce many, one is going to happen one at a time, because this is going to happen every month for the females up until they reach menopause. And then one cell is going to undergo meiosis to form a haploid ovum. Now there's a story of one becoming polar bodies. Don't go into that story. Just know that one cell is going to now undergo meiosis and then to form what? A haploid ovum. So one ovum is formed at a time. Only one. Unlike with the sperm where we had 4 haploid sperm cells, but with the female's ovum, you have 1. So how is that ovum now that is formed? So the ovum has a jelly layer from the outside. Okay, let's start from the inside. Inside, you have the nucleus. The nucleus contains 22 autosomes and 1 gonosome, which is X. Why is it X? Because females can only produce X. They can never have a Y. So it will have 22 autosomes and 1 gonosome, which is X. We know it's a female. Then the cytoplasm around the nucleus, see the cytoplasm right there, it nourishes this ovum. It gives it the nourishment that it needs. And then surrounding the cytoplasm, you have the cell membrane. Cell membrane. Now this cell membrane only allows one sperm to penetrate the ovum. Remember, the sperm is going to come with its acrosome wanting to penetrate here. So it only allows one sperm cell to enter. The fastest sperm cell will be the one that is fertilizing this nucleus here of this ovum. Then the rest of the sperm cells are going to go away, go back, go back, go back. So that is because of what? The cell membrane. And then the jelly layer protects the ovum against drying out. So this is your structure of the ovum. And now you know how it is formed. So you can answer a question using these 3 bullets on how an ovum is formed. And you can also be able to draw an ovum with its labels over here. Right. Then we come to a question. So this question here, fortunately for you, it has labels. So if it did not have labels, you were going to label it. Let us take a look at the labels. The diagram below shows sperm cell, shows a sperm cell. So there is your tail. You see it. You see the tail. They have just emphasized it by drawing an S tail just to show you that it is long. And then the mitochondria in the middle piece there. And then your head consisting of the nucleus and the acrosome. So you see. Now, state how the following structures are suited to assist the sperm cell to perform its function. The function of the mitochondria. How does it help the sperm cell? We've spoken about it just now. The acrosome. How does it help the sperm cell to perform its function? Acrosome, remember the one on the head, that is the enzymes, mitochondria, energy. So you must remember those things. And then sperm cells and ova are produced by gametogenesis. We've spoken about that. This happens when a diploid cell undergoes meiosis to produce a haploid cell called gametes. Now they are explaining to you what gametogenesis is. Then the question will be, tabulate 2 differences between gametogenesis in males and females that are not mentioned in the text above. So you must tell us how is oogenesis and how is oogenesis different from spermatogenesis. Right. So let us take a look at the answers. Mitochondria, the increased surface area for cellular respiration to provide energy. Or you can write for cellular respiration, very good also. The sperm needs that to have energy, to break down those ATPs, to have energy. And then also, yes, to provide energy for the sperm to swim. Remember that swimming fast, it needs energy for it to do that. Then the acrosome contains enzymes to dissolve the outer membrane of the ovum for the sperm cell nucleus to enter the ovum for fertilization. So you can mark also any 2 here. So those are the functions of, those are the structural suitabilities for the mitochondrion and the acrosome to help the sperm to perform its function. Then the differences between gametogenesis in males and gametogenesis in females. Let us take a look at those. Gametogenesis in males is called spermatogenesis. By saying that, you get a mark. Whereas in females is called oogenesis. And then it is stimulated in the males. It is stimulated by testosterone. Whereas in the females is stimulated by FSH, by the released by the pituitary gland, follicle stimulating hormones, because you need those follicles to form your ovum. Then In the males, gametogenesis takes place in the testes or in the seminiferous tubules, whereas in the females, it takes place where? In the ovaries. You see, we've spoken about these things. Now you are thinking about them deeper when you are differentiating them. And then gametogenesis in males results in 4 sperm cells being produced, whereas gametogenesis in females results in 1 ovum being produced. Then this process starts at puberty for the males, but for the females, the process starts before puberty or it starts at birth. All the follicles that they need to have for their lifetime will be produced at birth, but they will be activated when during puberty, but they are already there when they are born. So those are the differences between gametogenesis in males and gametogenesis in females. Right. Then we come to the most challenging part that you always complain about. The graph. You cannot interpret the graph, the menstrual cycle graph. This is a graph that explains to you what happens when during the menstrual cycle from day 1 up to day 28. So let us take a look. I want you to write down these words or these letters. FOLP is an acronym. FOLP. FOLP is the order of which your hormones are going to control this menstrual cycle. So we'll start with the first hormone. I will tell you this F will be your FSH. The O will be your estrogen. L will be your LH. And P will be your progesterone. Right. So if you know them in this order, FOLP. FOLP. Remember the FOLP. Follow the FOLP. We start with the F. That's where we start. This is our first hormone. You come to the graph and you check. Go where it says FSH. You see that, okay, this was day 1 when the person was menstruating and everything was under normal. So it can happen up to day 7, you see. So FSH will then be secreted by the pituitary gland. FSH, follicle stimulating hormone. Because the word says follicle stimulating hormone, you must go check on your graph what is happening in the ovaries. So this will represent what is happening in the ovary. You go check. The follicles are now developing. Follicle stimulating hormone is going to stimulate the development of the follicles. Then the follicles are going to develop and they are going to grow for a number of days. They are going to mature and undergo certain stages. And then once they undergo, you see where I've drawn the dotted lines. They are going to release hormone estrogen. So this is our second hormone. So this is our first. This is our second. The follicles are going to release the hormone estrogen. So this is where your second hormone comes. Now this estrogen, that is, okay, I just want to write it here. That is secreted by the follicles. Is gonna go where? It's gonna go act on the endometrium, on the lining of the endometrium, and makes the endometrium to become more thick and more vascular. So meaning it makes it more thick. So you start seeing, you see where I'm drawing the dotted lines now. The endometrium lining is thickening, is becoming thick. It is because of what? Now hormone estrogen is secreted by the follicles that were produced. And then on day 14, the pituitary gland is going to secrete hormone LH in high concentration. So LH, now we are on our third hormone. LH is therefore going to stimulate ovulation. What is the ovulation? It is the stage whereby a mature follicle ruptures and releases an ovum. So meaning that ovum is going to be released on day 14. That is called ovulation. That ovulation, you start seeing there on day 14, the level of LH, follow LH. On day 14, LH is high. Then, because it is high, it means in the ovaries, it brought about the rupturing of that ovum, of that follicle, to release an ovum. And then what is happening? Once it has done that, this ovum is going to form what we call a corpus luteum. So you see here, it's going to form what we call this yellow body. It's called a corpus luteum. Now this corpus luteum, once it is formed, it is also going to secrete another hormone, which is our fourth hormone. It is going to secrete progesterone. Now this progesterone, because once you see the yellow body, you go back to the endometrium and check. The endometrium is even more vascular. Why? Because this yellow body means, look at the blue line. The blue line shows you that after the yellow body was produced, progesterone goes high. Why? Because this yellow body is the one that is giving us this progesterone. So progesterone only start being secreted in more quantities by the corpus luteum. And you see it happening by the increase on the graph here. And you also see its effect in the endometrium, because you see the endometrium there, it is preparing for pregnancy. It is becoming more vascular and more thicker for that implantation. But if fertilization does not take place, look, the corpus luteum degenerate. It becomes small, or it disappears, You see it now becoming small. Then you know that fertilization did not take place. Therefore, the levels of progesterone is going to go down. You see, as the progesterone is getting smaller, look at the blue line, the progesterone is going down. Therefore, it means this person will undergo what? Their shedding, or there will be shedding of the endometrium, which is the menses, and then the cycle will start over again. So follow the FOLP method, and then you will see that you now know how the hormones work one after the other. And then the next diagram shows us the same FOLP with a different graph, but with the same interpretation. You know what is happening in the ovaries. You know what is happening with the body temperature. Then what are the pituitary hormones that are released? What are the ovarian hormones that are released? And what is happening in the uterus? All of these are intertwined. They are happening all at the same time. Now I want you to look at this. How do you know that a graph is showing you whether fertilization took place or fertilization did not take place? Just take a look at this corpus luteum diagrams here. Do you see it disintegrating or becoming small or disappearing? No, it is maintained. It has maintained its size. Therefore, even the level of progesterone is maintained. And therefore, look at what is happening in the uterus. The uterus is also kept. So meaning that pregnancy took place. This person is pregnant. Fertilization did take place because the progesterone is maintaining the pregnancy, is maintaining the uterine wall, and it is also kept constant because the corpus luteum did not disintegrate. It disintegrates when there was no fertilization. But if there was fertilization, therefore, it becomes maintained like this. So that is how you differentiate between the graph where fertilization took place or fertilization did not take place. So those are your graphs. I hope by now you know. Then, what is fertilization? Fertilization is the fusion of a haploid nucleus of the sperm with the haploid nucleus of the ovum. This is a very full explanation. You are not just saying it's the fusion of the sperm cell and the fusion of the ovum. You are saying the fusion of the nucleus of the sperm cell with the nucleus of the ovum. That is fertilization. Where does it take place? We know that fertilization takes place in the fallopian tube. So you see here, this is a sperm coming in to fertilize the ovum. So once that ovum the sperm has met the nucleus of this ovum, then we know that fertilization has taken place. After fertilization has taken place, a zygote is going to be formed. Now, this zygote is diploid. Diploid, why? Because it consists of 23 of the males plus 23 of the females. Remember, sperm, 23, ovum, 23. So it is now haploid. Then, here, let us follow the development now of this zygote. The zygote is going to be formed by the nucleus of the sperm cell, and then it's going to form two cells. It's going to undergo mitosis now. Then form 4 cells body, 8 cell, 16. Then once it forms 16, we call it a morula. And then it reaches a stage where it has a halo. It's a halo, a ball of cells with a halo in it. Then we call it a blastula at that stage. And then you see it is developing where? Inside, follow the arrows. It is developing inside the fallopian tube. So once it has reached the stage of becoming a blastula, look what happens. It embeds itself on the endometrium. Therefore, when it attaches itself on the endometrium, this, we call it implantation. So this is just a zoomed out picture that shows you how implantation takes place of this blastula. And then the blastula is the one that is going to grow into an embryo until it becomes a fetus. So this is fertilization, the development of the zygote, and implantation. So you've done all those 3 processes in one diagram. Then, after that implantation, the fetus needs to develop. So what is happening? What is surrounding the fetus? What is happening from it being a blastula? How did it develop? So let us follow what is happening here. We have the chorion, which is the outer layer that is covering our fetus. We can say it's a fetus now because yes, it's at a stage that is at a fetus. So that covering will therefore grow into finger-like projections. You see these projections. We call it, when it comes here, remember it is the chorion here. And then when it becomes the finger-like projection, we call it the chorionic villi. The chorionic villi are finger-like projections of the chorion, which projects into the blood spaces in the uterus of the mother. So this here, we will say, is the mother. Right, so this finger-like projection enters into the blood spaces of the uterus of the mother. So this is the uterus. Then it provides maximum contact with maternal blood, and also it forms part of the placenta. So you see, you have done now the chorionic villi coming from the chorion, which is the outermost layer. And then you come to the umbilical cord. You see the umbilical cord? Umbilical cord here. Here's the umbilical cord on the baby, coming from the navel of the baby there. The umbilical cord is a flexible cord containing 2 umbilical arteries and 1 umbilical vein. So you see there are 3 things there. 2 arteries, 1 vein. The arteries carry carbon dioxide, while the umbilical vein carries nutrients, meaning the baby will receive nutrients through the umbilical vein and will carry out the carbon dioxide through the arteries, the umbilical arteries. Then you have your amnion. The amnion secretes amniotic fluid. So you see the amnion is this one, is this sac. You see? The amnion here secretes amniotic fluid. So here's the amniotic fluid inside here, and protects the developing fetus. So it protects the developing fetus and it secretes that amniotic fluid. That amniotic fluid then, what is it going to do? It is going to act as a shock absorber to protect against temperature changes and protects the fetus against dehydration. So this fluid that the baby is swimming in is going to protect the baby against shock and also temperature changes, and also protects the fetus against dehydration. So that is the umbilical, the amniotic fluid. And then you have your placenta. Now you see here is your placenta. Placenta is for gaseous exchange between the mother and the fetus, providing nutrients to the fetus, and it secretes progesterone. So this is your developing fetus. This is how it's going to survive for 9 months during this gestation period for a human being until it is born. So all of these layers play a very important part in the developing fetus in the uterus. So these are the questions that come from KZN prelim. So let us take a look at this one. The diagram below, remember, you can also start by studying this diagram and check where can you put the label, what can P represent, what can Q all together represent, what is happening here at A. I see there is blood vessels of the fetus, it's moving in the direction of the fetus, then this is moving in the direction away from the fetus. Then you must remember what is happening. Then this whole of thing is the placenta. You see, you are studying what is happening there. The diagram below represents the relationship between the blood systems of the fetus and that of the mother. The arrows indicate the direction of blood flow in the blood vessels. Now, identify structure P and the extra embryonic membrane. What is the extra embryonic membrane? We spoke about a membrane just shortly. So your structure P will be chorionic villi. Villi, why? Remember, finger-like projections, and they are projecting into the endometrium of the mother. So they form part of the placenta also. Then the extra embryonic membrane, so that extra embryonic membrane that will be covering the fetus is called what? The chorion. Remember the chorion that is giving us the chorionic villi there. And then give two functions of the placenta. So this placenta right here, what does it do? We've spoken about that. You can give any two. It acts as a microfilter, preventing harmful substances from reaching the fetus. Remember, it has those blood vessels. It also produces antibodies. It secretes progesterone during pregnancy, which maintains the endometrium. So progesterone is going to come even from the placenta to maintain this pregnancy, because we don't want it to shed to become a miscarried or to become a miscarried embryo. Then immunity is transferred from the mother to the fetus through the placenta. So you can give any of those two functions for the placenta. Then I would like for you to explore these further questions. You have the worksheet. Explore these further questions on your own based on the placenta, implantation, and the development of the fetus. Right? So we can move on. Now, this is the question that came from the paper that we've just written. Now, if you take a look at this, okay, let's just write, this is our label D here. This is our label D. If you take a look at this diagram, this is just the diagram that we've just spoken about. It is the diagram showing you the development of a fetus together with all the layers of the placenta, the uterus of the mother, the chorion covering, the amniotic fluid. You see, you can tell a story even before you go to the questions. Now, let us go to the questions. It says, identify label B. Then you know you must tell us what is B. Right. Name two functions of A. This A is the fluid that we find here. Describe how structure C is formed. Structure C is this one, your placenta. Then tabulate two differences between the composition of blood found in the blood vessels in D. So the blood vessels in D here. So what is the composition of blood found there? So let us take a look at the answers. So here, you try to provide the label for B. What is B? Chorion. Were you correct? I hope so. Name the two functions of A, which is your amniotic fluid. We said what are the functions there? Shock absorber or protect against mechanical injuries. Temperature regulator, prevents dehydration, or it is a medium for the fetus to move in. Remember, the fetus will change position as you go for sauna. You will see the fetus moving and changing. So when you see this in a marking guideline, when it says mark the first two only, you were asked to give 2. So you have given 4. If your first 2 are incorrect and the last 2 are correct, you are going to lose marks because then the marker is going to mark only the first 2. So be careful of writing many answers when you are asked for 2, because then you might start with the wrong ones and end with the correct ones and then lose marks. Then 3.3.3, describe how structure C is formed. So how is the placenta formed? The chorion forms, then the chorionic villi is going to form, and then it attaches to the endometrium. You see, from the chorion to the chorionic villi, and this chorionic villi then attaches to the endometrium, which is the wall, the inner lining of the uterus of the female. So that is how the placenta is formed. So all of that, the baby and the mom, it forms the placenta. Explain the endocrine role of part C during gestation. So part C, what does it do? What does it help with during this process of gestation or during pregnancy or during the process where the baby is kept inside the uterus for development? That is your gestation period. Remember, it is going to secrete progesterone. We've just spoken about that. It is going to secrete progesterone, which further increases the thickness of the endometrium because we want to maintain this pregnancy. We said not only the corpus luteum is going to secrete progesterone, but now we are also adding that even the placenta is secreting this progesterone that is going to help maintain pregnancy and then making it more vascular and glandular, so it becomes more. Then tabulate 2 differences between the composition of blood found in blood vessels in part D. So blood vessels in part D, we said this is our D, which is your umbilical cord. Then, so the umbilical artery and the umbilical vein, because they are 2 blood vessels that are found here. Remember, then we said this one, umbilical artery carries deoxygenated blood from the fetus to the placenta. Your carbon dioxide is going to go out. It's going to excrete toxins. And then umbilical vein carries oxygenated blood from the placenta to the fetus. So this one is going to come from the mother to the fetus. And then the umbilical artery carries toxic substances from the fetus to the placenta. So the umbilical artery is part of the excretion from the baby. And then the umbilical vein carries nutrients. It brings in good things to the baby. So the vein brings good things, oxygen and nutrients. But the umbilical artery, it takes out carbon dioxide and it also takes out toxins. So those are the 2 differences. If you may forget them, just remember them in that way. Artery takes out. Arteries takes out away toxins, away carbon dioxide or deoxygenated blood. And then vein brings in oxygenated blood. It also brings in the nutrients. So I hope after answering this question, you are now confident to even apply this question, or to answer this question when you come across a question that is asking you about implantation, fertilization, and the development of a fetus and gestation. Because most of the time, you just skip through this topic here. Right, and then we come to the second big topic that we said we are going to look into. So we will just look at the highlight some of the parts that you need to go through for this topic. So the topic is human responses to the environment. So how do humans respond to the environment? What does the exam guideline say? It says you must know the structure of the brain. Okay, let's have all our information from the exam guideline. All of it was taken from the exam guideline. So the exam guideline expects you to know the brain, the structure and functions. If you see a brain, you must know which part is the cerebellum, medulla oblongata, hypothalamus, corpus callosum, and their functions. What are their functions? Then you must know the spinal cord, the structure of the spinal cord, and describe the reflex action using a diagram. Then neurons. You must know that neurons are specialized cells which connect to the brain and the spinal cord to all other parts of the body. And we have 3 types of neurons, and you must know their functions. Those are your motor, your sensory, and your interneuron. And you must be able to tell which one is which. looking at the diagrams. Then you must know the human eye. You are still responding to the environment through the eye. Know the structure and functions and the parts of the eye. Accommodation, which is the adjustment of the shape of the lens to see objects clearly, whether they are far away or close to the eye. So that accommodating the distance, whether it's near or far, that is accommodation. So you're changing the shape of the lens to accommodate that. So you must remember it's the shape of the lens to become more convex or to become less convex. We are talking about accommodation. Pupillary mechanism regulates the amount of light entering the eye by adjusting the size of the pupil. So you see, these are two things. The accommodation deals with the shape of the lens, but pupillary mechanism deals with changing the size of the pupil. So it becomes, the diameter becomes wider or it becomes less wide. So then you must also know the visual defects. Then you must know the human ear, the structure and the functioning of the parts of the human ear, the process of hearing and balance and hearing defects. I know here with balance, most of you don't even consider looking into balance, but there is a way to go about shortening it or making it simpler for you. So all of these carries 54 marks of your paper one. So it's quite a lot. Please make sure that you collect a lot of marks. Your structure and function. Structure and functioning. Structure and function. Please, that is where everything starts. Once you start with the structure and functioning, all other additional information will come in as you understand the functioning of a particular part. Right, the nervous system consists of the central nervous system, which consists of the brain and the spinal cord. We've spoken about that. And then it also consists of the peripheral nervous system. Peripheral nervous system has the sensory pathways and the motor pathways, which will further be divided into the somatic or the voluntary nervous system and the involuntary nervous system. And the involuntary nervous system brings the sympathetic, it has the sympathetic and the parasympathetic division, the one that allows you to react and the other one that allows you to come back to normal or to restore you to normal under such a reaction. So let us take a look at the structures and the functions. So if you take a look at the central nervous system, the CNS, we said it consists of what? The brain and the spinal cord. So this here will extend to become the spinal cord. And as you can see, the brain and the spinal cord, those are central to your body. So you cannot remember, you cannot forget that. And then the peripheral nervous system is the one that is coming where? On the side. So you see, and it is branches to the motor, then the voluntary, involuntary, and the parasympathetic and the sympathetic, the flight or fight, and the one that brings you back to normal. And then the reflex arc. You must know when they are asking you about the reflex arc and what is a reflex action. Reflex arc is your pathway. This is where you are tracing where it starts, where this nerve impulse is gonna start, and where it's gonna go through, until where. So you are tracing the path. It's an arc. But the action that's gonna come there, once you react, the reaction is called what? Reflex action, because now you are doing what? You are acting. So the action is the acting part by response, and then the arc is the pathway. Right. So what are the parts that you need to know of? We're not going to look at the functions. I want you to go look at the functions. But when you see your brain, you must be able to label it. So which parts can you label there? This is your the cerebrum. How I would like you to call it is to say is the cerebrum. Why? Because we have another part that have a name that sounds similar to that one, but it is a different part. So the cerebrum is the largest part of the brain there on top, and then you have the corpus callosum dividing the two hemispheres there, and then you have your hypothalamus. Remember, when you're doing negative feedback, you talk about the hypothalamus, and then you have the cerebellum. Now the cerebellum is the one that looks like a tree. You see, it looks like a tree. It is the one that is responsible for balance, right, and coordination. So, and then your medulla oblongata for breathing. And then you have your pituitary gland or the hypophysis. And then pituitary gland secretes so many hormones. This is a very important gland in our bodies because it regulates most of the processes that are taking place in our bodies. And then leading to this one, we said here this one will be our spinal cord. Right. Then our somatic nervous system of the peripheral nervous system and the autonomic nervous system. And we said the autonomic nervous system has the sympathetic division and the parasympathetic division. And you must know what is happening during a flight or fight response. How are your body parts reacting? Your eyes, your pupil dilating, your heart rate, your everything that is happening in you during a flight or fight response. You must be able to describe that. And then when we come to a reflex arc, we tracing the pathway. Then you must know that it starts with a sensory receptor. Why do we say receptor? Receptor to say we receive. We receive the stimulus. This is what is going to bring the stimulus into your body to be interpreted as what. And remember, reflex action is a quick, rapid response. There is no thinking. The brain is not involved. If you take a look at this path here, you see it only goes through the sensory receptor through the spinal cord up until it comes to the effector. So you respond quickly because you don't have time to think about what your response should be. Hence, the action is going to be rapid and very fast. Right. So we start with the sensory receptor here. Your stimulus is going to be received here, and then that information or the nerve impulse is gonna travel through the sensory neuron. The neurons only transmit impulses because it's an electrical system. So that impulse is going to go through the sensory neuron. And the sensory neuron is this one, you see, on the dorsal root of the spinal cord. So this is the dorsal root, this is the ventral root. It's going to go through the sensory neuron, and then inside the spinal cord is going to join or is going to transmit those impulses through a synapse that has neurotransmitters to the interneuron. So that is the neuron that is in the middle. And then from the interneuron, they are going to be transmitted, the impulses are going to be transmitted to, so this is our spinal cord, they are going to be transmitted to the motor neuron. The motor neuron is the neuron that is underneath. And then from the motor neuron, it's going to go to the effector. Why do I say, I like to say effector is where you're going to feel the effect and then respond. Sensory is where you're going to sense the stimulus. So you see, start with the sensing and then it ends with the effect. So where you're going to feel the effect, the effector. So then you will respond accordingly. So that is basically the short and sweet of your nervous system. So how do you differentiate between the three types of neurons? You know, your sensory neuron is the one that is your first one. So your sensory neuron is neuron number one. It's the one that is going to start. You see, when it starts here, it does not have terminals, it does not have dendrites. So it starts from the receptor cell. And look at the direction. Look at the direction of the impulse. It's coming from the receptor down through the axon, covered by the myelin sheath. So you see this sheath that is covering here. It is the myelin sheath. And look, the cell body is on the side. So that's how you know that this is a sensory neuron. And then it's going to extend to the axon. At the end of the axon, you find what we call terminals. Now, these terminals are the ones that are going to join the dendrites of the next neuron. And the next neuron after the sensory neuron we know is the interneuron, or is the one that is in the middle, is the connector neuron. So you see in your head, you take these terminals of the axon of the last and then you put them here. They join what? Through the neurotransmitters of a synapse. They join and transmit that impulse from the sensory now into the inter. The inter will receive those transmitters by the dendrites. So the first thing that will receive those impulses are the dendrites. And the dendrites are then going to transport, look at the direction also, transport those impulses through the cell body to the axon. And remember, at the end of axon, we find the terminals. Now, these terminals of the interneuron are going to join what? The dendrites of the third neuron. So this is our second. This is our third. So they're going to join here through the neurotransmitters. Remember, from here to here. Through the neurotransmitters here, the impulse is going to get into the dendrites of the motor neuron. And look at the motor neuron. Okay, here the cell body is in the middle. If you have forgotten, remember, interneuron, middle is the connector neuron, connecting the two neurons, the sensory and the motor. And its cell body is in the middle, there. And then here with the motor neuron, the cell body is within or in the dendrites. You see, in the dendrites here. And therefore, that the direction also of the impulse is going to go through that cell body, through the axon to the terminals. And then from the terminals here, they're going to go to where? The effector, where you are going to feel the effect. So that is the direction of the impulse through a reflex arc. Then that is how you also know your functions. Right, please go through the questions of the brain, because you have the workbook, you have the worksheet to work through this. Go and study the functions of the different parts there. Right, so those are the answers of some of the questions that we are going to go through. I want us to cover the content. And then here, this was a question coming from the Eastern Cape asking you, by looking at these diagrams, are you able to tell which one is the sensory, which one is which? So you see this, the cell body is in what? Inside the dendrites. Which neuron will this one be? This one, the cell body is on the side. Which neuron will that be? So you must be able to answer those questions. Then identify those and look at the structures and functions. And also here you have your question paper from 2022 that was asking you about the reflex arc. So if you are able to tell us that from here you have your sensory, your interneuron, and your motor neuron, you will be able to answer any question that is coming after that. So please go through this question as well. Right, so the structure of the eye. The structure of the eye, how are you going to look at the structure of the eye? Please start from the outermost layer and get into the innermost layer. Remember how we did it before the prelim? You start from the outermost parts and you come to the innermost parts. And then with the eye also, you must be able to tell us how does the eye accommodate or vision for near and far vision. So this is what you can use to remember it. Please write it down if you did not get a chance to write it down the last time. For object that is less than 6 meters away, so this is near. For object that is more than 6 meters away, this is far. Write these letters: CC, SS, TD, LM. What are these? This is telling you that the ciliary muscles is going to help you remember the process of what is happening during accommodation for near vision. Ciliary muscles are going to contract. If you can look into Mind the Gap, you are going to get all these steps. So please go through your Mind the Gap and check all these steps. But trace what CC is, SS, TD, LM. Then you have all your answers. And then for an object that is far away, you have CR, that is your ciliary muscles are going to relax. And then your sclera is going to be pulled backwards, and then the suspensory ligaments are going to be taut. Now this one is just an additional information. And then the tension on the lens is going to increase, and then less light is going to enter the eye. So the lens becomes less convex. Therefore, you see, so please make sure that you remember these, and you apply them to here. You see this presentation, you have the T's and the I's and the L's showing you what CR is, ST is. So please go check your accommodation in this way. The eye is not only responsible for accommodation. Remember, it is also responsible for the vision in bright or dim light. That is pupillary mechanism. It adjusts the size of the pupil based on how much light is entering the eye. So it's controlling that. So how are you going to remember the process? Right, again, for pupillary mechanism, CC, RR, PC, and LL. What is it? The circular muscles are going to contract. You see, this is your first CC. And then the radial muscles are going to relax. Radial muscles are going to relax. The pupil is going to constrict. And then less light. Yeah, for LL, right, less light is going to enter the eye. This is during bright light, during the day. And then during the night, in dim light, it is CR, RC, PD, ML. So you want more light to come in. What is going to happen? Your ciliary muscles are going to relax. So ciliary muscles relax. Unlike here, ciliary muscles contract. You see, you have CR now. Then the radial muscles are going to contract. It's the opposite of what was happening above there. The radial muscles are going to contract, and then the pupil is going to dilate here. Pupil dilate, and then more light is going to enter the eye. This is during dim light because you want to see more. So I would like for you to go through all of these processes that we have done today to help you remember the parts that were very challenging when you were writing your preliminary examination. And I hope it puts you best to collect the 48 marks and the 54 marks of these two particular topics. Please go through the worksheets that is provided to you. Please don't cheat. Make sure that you practice. All the diagrams that you have, all the diagrams that you see in all the question papers, label them. Start by labeling. Make sure that you know the functions and then answer the questions. And please make sure that you master the skill of drawing a graph, because that's where you're going to get other marks for you to perform better in life sciences. So I hope to see you again next week when we are doing the challenging topics that you came across in paper 2 for life sciences. Thank you so much for watching this lesson. I will see you again next time.